video. Hello, hello. I'm going to turn this up. That way if somebody says something, we can hear them. Okay. I'm going to go to Moodle. I do need to share my screen.
If we're sitting in, do we have to get on Zoom?
No, you don't have to. You don't have to. All right, let's see. This should come online in like 4 minutes. Okay. I want this one. And, oh, stop it. I think I just clicked the pen. I want to annotate. Okay. I do need the clicker because we need attendance. How many things do you have to log into before you can teach? Let's see. Should start running the cloud. Got the chat up. And let's go to this one. Okay. And then attendance. So I'm going to minimize the chat and make this big and start attendance. Okay, so we get a chat question. Oh. And Ross, you need to work on getting that clicker working. I don't want to make the chat that big. Okay. All right. If you need help, let me know. I'm happy to help. Okay. Now, why isn't that loading? Can y'all access the discussion questions? What are we listening to out there? I'm just gonna dance. Y'all have no idea what I'm dancing to because you're at home. Yes, you can access it, Rebecca. Okay, let me just try again. Whoa. Chapter 3, discussion questions. Okay, for just some reason, I guess it just didn't load. Oh, but that's a PPT. It needs to be a, it needs to be a Google sheet, not a PowerPoint. Oh, Dr. B made a mistake. I'm gonna give y'all a minute. I'm gonna have something else for y'all to do and I'll fix it then. So you probably can't write on it right now. Okay, so class is open, so the Respondus thing should be open, and I'm gonna unhide it because it should be unhidden by now. So let me just do that. Share topic. Hey. Okay, so everybody go to Moodle. I want to show you how you are going to take your exam. Something in the chat. Hello. Hello, Jennifer. All right, so everybody should be checking in on their iClicker for attendance, and they should be going to Respondus, this Respondus topic here in Moodle. So I'll make that big. It's right under course materials, and then you have Respondus exam materials. So anytime you have an exam, right, in chemistry, usually we have some kind of exam resource that you're going to have. And so in this case, for this exam, it'll just be the periodic table. So you will get a periodic table, right? We are going to use Respondus because we cannot all fit in the classroom at the same time. Taylor, reload your Respondus, your Moodle. Me either. Why is it restricted?
Okay, refresh, going to refresh.
I got it now after refreshing. Okay. Okay, good. Good. Okay. So your exam resources periodic table. You have a video on how Respondus works. It's going to be a lockdown browser so that when you open the exam in Moodle, you can't go anywhere else. Okay. You can download Respondus to your own computer, or you can use a campus location that already has Respondus. These are the locations where Respondus is already downloaded. All you would have to do is show up at this location and open any browser, go to Moodle, and click on the quiz, and it will automatically open your, not quiz, your exam in Respondus. Have y'all have used Respondus before? Okay. This was not the case with my other class, which was interesting. If you're on campus, do we have to sign up to be in room? No, Jennifer, only if you want to be in this particular room do you have to sign up. Okay, so I'm gonna show you that in just a second. So these are the instructions on if you want to download it yourself. And then here's a practice exam. Why is this one hidden? I want this one to be unhidden. Hold on, I'm gonna unhide it. Edit settings. Let's see. That was fine. I keep forgetting where. Hide here, show. Okay, it should show up now. Ooh, went too far. Here we go. All right, so this link at the bottom, if you just refresh your window, will take you to an Excel spreadsheet. How is there a surroundings check without? You have to have a camera on your laptop if you take it on campus. You have to have a camera. Okay, so this is the link that will take you to the Excel spreadsheet. There are 32 slots available. If you want to take it in this class, you need to sign up to make sure you have a spot. So it'll take you to one, and then just put your name on the list. Okay? And if you want to like skip down a few so nobody overwrites yours, that's fine with me. So just skip down. It doesn't have to be one after another. I know because Excel can kind of go crazy. So just kind of pick a random one and put your info in. It doesn't matter what number you are. Yes, ma'am.
Should I do that too in case the setting doesn't let me put them straight down?
No, you're good. You're good. You're good. Okay. Let's see. So, oh, where's it over here? Okay. So everybody's signing up. What I want you to do is I want you to take this practice exam with whatever computer you are going to use. This Respondus practice exam, you can take it as many times as you want. Does not matter, because I want you to be able to say, oh, I was going to use my laptop, but I decided to use my desktop at my parents' house. Whatever. I want you to be able to try it again. Okay? So it's going to be very similar to the practice in structure, right? Each question is going to be on a new page. You know, all that kind of stuff is going to be really, really similar. So I want you to go through the whole thing and try it at least once on whatever computer you're going to use so that we can kind of cut down on possible technology things. But it says 3 attempts. Oh, this is how many people have already attempted it. It's not because you're seeing it from my perspective. My perspective tells me 3 students have attempted it. Excuse me. Okay, so you can open it as many times as you want because the grading method is the highest grade, but you don't get a grade. There's no grade. I hope I did that right, that there's no grade. Right, it is. The material is the periodic table for this one, but like next chapter, you'll need a codon chart because you'll have to be able to translate from DNA to mRNA to protein. Well, if you don't have a codon chart, you can't do that. I'm not expecting you to memorize that. So I'm telling you, this is how you'll see those resources. Okay, all right. So we good on Respondus? Any questions on that? Okay, so I have to apologize. It's on my list, actually, apologize to students because I got ahead of myself last class period, and I was in section 3, and y'all hadn't done section 3 yet. So this is one thing that I'm having to get accustomed to. I'm having to get accustomed to because I pre-record all the lectures, so I'm like way far ahead. And I kind of have to come back and remember where y'all are at. So I apologize for that. So I will try not to do that in the future. That was my bad. So I want to, mine isn't making you use Respondus to take it. Really? Let me see. Let's see, hold on. Where's my, it's not making Respondus. Oh, hold on. We're gonna fix it. Okay, I will fix it while y'all are working on something because that's the 2 things I have to fix. So I'm gonna check it, Respondus, and I'm gonna check the PPT to Google slide. Okay, because I'll have a minute while y'all are doing something. And I will check it. Okay, Taylor, Emily, everybody, I'm gonna fix it because it's not up here and it should be up here. So I'll fix it. Okay, so what I want you to do is to go to Moodle, and under section 1, chapter 3, there's a little peptide activity I want y'all to do. We talked in the lecture a lot about you have to do some cloning. You want to express a protein. You want to actually do these things. And it's great that y'all got kind of like the big concept on how it's done. But when you sit down in lab and your PI tells you, I want you to clone this gene in, you're like, okay, I don't know what I'm doing, right? So I want to give you guys a little bit of a practical experience on what you would do. Now it's not going to be perfect, but it's a good replication of what you would kind of have to decide on and things you would have to do if you were actually given a cloning project. And this is a lot of times a very first step in a lot of scientific research. So if you pull this up, it's cloning a peptide. So as a part of your undergraduate research, you are tasked with cloning, expressing, purifying, and biochemically testing a small peptide. We're going to pretend it's called peptide X. You're told the sequence of the DNA that codes for a particular part of the protein. We're calling it a peptide because this is really short. Most of them are longer than this, but this is relatively short. So this is a fragment of genomic DNA. Usually we know we have like promoters, we have introns, we have all that kind of stuff. What I'm telling you here is there are no introns in the fragment. So what I want to be able to do is to cut out the important part and put it into a vector. How do I do that? What kind of enzymes do I need to make that happen? Right, I've got my bacterial plasmid and I've got my, let's say this is a human genomic DNA sequence. What do I need to do? What's my general next step? We talked about specific enzymes that we would use. What would we use? So what is EcoR1? What's the general classification? EcoR1 is an example. Endonuclease restriction enzyme. Good. Yes, exactly. So we need some restriction enzymes. What do restriction enzymes do? They cleave what? DNA. And my plasmid is, guess what? DNA. My fragment is DNA. So I want to cut both my little piece of genomic DNA and my plasmid with the same restriction enzymes so that I can insert my peptide into my little plasmid. Okay? Well, that's great. Do you know how many restriction enzymes there are? There are books of the restriction enzymes, books of them. So how am I going to choose which enzyme to use? Thankfully, your teacher was really nice and gave you a short list. But in general, what would I need to make sure? Whichever one is contained in both, right? So I need to have a restriction site here, and then I need to have that same restriction site in the multiple cloning site of this plasmid, so the MCS, so it's got to be over here. So that's one. What else do I need? Another one, right? I want to cut out the important bits and stick it in. So I have to cut both sides of my peptide, my genomic DNA piece, and I have to cut twice in the plasmid and then ligate in my piece of interest, right? So I'm giving you the piece of interest. In order for a restriction enzyme to work, you have to have a palindromic sequence. So remember when I was showing you those pictures of the enzymes cut sites? You know, there were all kind of different examples. Well, here are those examples. And I want to switch over to the one that I can write on better. So I'm going to switch over to this one. Fit with. Okay. So maybe I can even zoom in even more. What's my maximum? Whoa. Okay, I don't need the maximum. All right. So I have different restriction enzymes, and it's telling me that this is the restriction sequence right here. What does this mean? G/AATTC. Are you cutting single-stranded DNA? No. We're cutting, yes, Ross, you're right. We're cutting double-stranded DNA. I'm only giving you the template sequence at the top, 5' to 3'. But there's another sequence, right? A, T, C, A, so on and so on and so on, right? So I'm really cutting double-stranded DNA. How am I going to do that? EchoR1 has this cut site. The slash here tells you where it's cutting. So for EchoR1, I have on one strand GAATTC. But in order for EchoR1 to cut, it has to be palindromic. So check this out. AATTC. So if I read the bottom strand backwards, it reads as the top strand, GAATTC. That's the characteristic of a restriction enzyme site. So what does that slash mean? That's where it's going to cut. So if it's going to cut here on the top strand, where is it going to cut on the bottom strand? After the second A. So this is how we draw the cut site. Okay? So what I want you to do is to find 3 6-letter palindromes that are in that top sequence. Okay? There are 3 of them. I want you to try and find them. So I'm going to give you a few minutes to try and find them. And in the meantime, I'm going to fix all the things that I didn't do right. And I'll leave the chat up in case y'all run into problems while you're doing it. Okay. I want to do the discussion first. don't think I brought my cell phone. Dang it. Sent to the, oh, I can do that. Oh, let's do my iPad. So, Apple. Do that. There we go. Okay. All right, so. Do y'all want me to leave the blank one up so that y'all can check your, like if you want to use it as a study tool where the questions are there, but they're blank, then you can check yourself? So I'll do that. And then I'll duplicate. This one. Okay, problem 1 should be fixed. Problem 2. Here we go. That's it. Okay, now your second problem should be fixed. Will there be one uploaded after this that has been graded containing the right answers? Christian, are you talking about the discussion questions? Christian, are you talking about discussion questions? Okay, yeah. So here, this one, you can't edit it. You can just see it. This is the one that you guys are going to work on. After we do this peptide activity, that's what y'all are going to work on. And then that's the one I will go back and check. And I'll go back and check it on Monday. I know it's a holiday, but I'll go in and check it on Monday so that y'all have all the answers and you know what they are before our exam on Wednesday. Does that make sense, Christian? Okay, are y'all close to finding it? Yeah, y'all got it? Okay, so tell me, what are the three sites? And I'm not gonna go through and draw all my palindromic, all my complementary, but what do you got? Where do we start? Well, I guess I'll just do what I got and you tell me if this is what you got. So I got, starts right here and goes to the T. That's one palindromic site, okay? Your next palindromic site is your sequence of 3 C's and 3 G's. Yeah? Okay, and then I got my, here, starting with the G and going to 2 C's. Yes? Okay, what do you notice about those 3 palindromic sites? Maybe having something to do with a recognition sequence of some of the restriction enzymes. Did anybody notice that they match? What's the first one? SAC1, SAC1. Call it SAC1. I don't know, maybe other people call it other things. But the little abbreviations don't always make sense. Okay, the next one. SMA1. Yes, Ross, good. And then the last one, BAMH1. Yeah, good. Okay, so when I'm looking at this sequence, what I want to do is I want to find out which restriction sites are in my little piece of DNA, my little it's not template, but the genomic DNA. So is it in genomic? Is a question mark, and then is it in the plasmid? Because it has to be in both, right? So which ones were in genomic? We said SAC1 is in genomic, SMA1, and BAMH1. So now I need to go and I need to check my multiple cloning site on my plasmid because I don't want to just put it anywhere. I don't want to interrupt my antibiotic resistance gene. I don't want to interrupt my origin of replication, right? You can't just stick it anywhere in the plasmid. You have to put it into the right place. So which multiple cloning sites are also on the plasmid? We have SAC1 and we have BAMH1. What else you got? You have EchoR1. Those are the only 3 that were on the site, the multiple cloning site, right? So the 2 that we're super interested in using are this one and this one, right? Now, that means I don't want to use SMA1. That's one reason why I don't want to use SMA1. Can anybody else tell me the other really, really important reason why I don't want to use SMA1? You would cut your genomic fragment there right in the middle. You would split your peptide. It's going to be non-functional. You would just shoot yourself in the foot right there before your experiment even started, right? So be careful that your restriction sites are flanking your peptide sequence, not inside of it, but flanking it, right? Okay, so now we know we can use EchoR1 and we can use, what was the other one? No, we're using BAMH1 and SAC1. Sorry, BAMH1 and SAC1. Okay, this worked out super nice because you want to put genes in the right orientation as well. Okay, so do you notice up here where we're going 5' to 3', right? We have 5' to 3'. You want to insert your fragment going the right direction, right? So that all the cellular machinery that normally does all the normal jobs will do it in the right direction. So you want to make sure that SAC1, where do you want that to cleave, 5' or 3' of your fragment? Where would you want it to go? Would you want the SAC site to be 5' or 3' of your genomic sequence? You want it to be 5', and whoa, look how awesome that is. It is. Hip, hip, hooray, right? And then BAMH1 is 3'. And if you come down here, look, BMH1 would be 3'. So this would be your 5', this would be your 3', and your plasmid's going in this direction, so you're good. That's like magical. Never happens that easily, promise. But it's a good little introduction on knowing all the different things that you need to get it in, right? Okay, so any questions? Oh, the other thing I wanted to say, right? Now, you've inserted it in, you've cut your 2 pieces, you put your pieces together, you put some ligase that'll seal the double-stranded breaks, right? Now, what do? You do what? You have a piece of DNA, theoretically, it's this plasmid that has an extra insert right there. What do you do with it? What do you do? Well, no, now I do want to make more of it. How am I going to make more of it? I'm going to what? Transform it. Somebody said it on there. Transform it. That's exactly right. We're going to transform it into some bacteria. What are we going to do with those bacteria? We're going to let them grow a little bit so they have time to take up the new plasmid. What do we call that new plasmid now that it has an insert in it? It's not just a vector. It's now a recombinant. Good, good, good. It is recombinant because we're combining things, right? We're combining maybe it's a human gene and a bacterial plasmid, right? So it's recombinant DNA. All right, so we take this plasmid, we've ligated it, we've given it to bacteria, they take it up. Now what? My goal of my experiment is to clone it. We cloned it, we put it in, express it. Ooh, I'm not there yet, right? So I took this and I put it in bacteria. Did every single bacteria get this plasmid? Do you think it worked perfectly for every single plasmid that was in my test tube? No. So you need to plate these bacteria that are growing with this vector inside of it on something with antibiotics. So maybe this is ampicillin resistance gene, and this is tetracycline resistance. So I can plate these bacteria on a plate that has, so it's media with ampicillin. A-N-P, I don't know how to spell ampicillin. A-N-P-I-C-I-L-L-I-N, something like that, close enough. That has ampicillin, that has the antibiotic. So only bacteria that have this plasmid are going to grow, right? So we're specifically selecting for them. You could also put inside of that media XGAL. If there's no insert, if you put everything together, but for some reason maybe your ligase didn't work, it got too hot, you didn't do your experiment right. So all the bacteria got this vector, but the vector doesn't have your insert. Well, if you put XGAL into your plate, if this LAC-Z gene is transcribed properly, then your colonies will turn blue. But here's part of the LAC-Z gene, here's the other part of the LAC-Z gene. If I insert a big old stretch of DNA and separate those two parts of the LAC-Z gene, so now I have a huge space here, the LAC-Z gene won't be transcribed properly. So it will no longer be able to break down XGAL, and the colonies will be white. So you want white colonies that, if I could write, that grow, right, on AMP plus XGAL, right?
So you're doing a double screen. You're saying, did they pick up the plasmid? And does that plasmid contain my gene of interest? You'll actually sequence it and definitely check, but you don't want to waste your time on something that doesn't have an insert. Cool? Isn't this cool? It's just like so amazing that we've hijacked all these systems. We didn't invent any of this. Evolution did. It's so cool. All right. So that's my little, I thought y'all kind of needed a little extra in-depth about plasmid construction. I thought that kind of would help y'all. And so I want y'all to work on the discussion in your groups for the rest of the time. If anybody has questions, I'll take questions now. And if somebody would go check and see if they can type on the Google sheet now, that would be awesome. So it should be this one. Fill this one in right here. Will this be similar? So Taylor, this will be on the test. I am not going to give you a test and say, it's one question, how would you create a recombinant vector? I'm not going to do that to you. I am going to ask you questions that depend on your understanding of the process. Okay, you were able to add a text box. Crystal, that's great. A text box would be good. So this is just one of those things that you need to understand the concept of so that you can answer questions. But I'm not going to make you do it. I'm not going to make you design a vector in an exam. I'd make you do it in the lab though, if possible. But does that make sense, Taylor? No, you don't have to turn this activity in. You have to turn it in. This is to help your understanding.
Can you explain how, at the very end of next generation sequencing, how you're figuring out which?
Yep, let me pull up an image.
Like I understand for the gene chip one.
Okay, so the question is about next generation sequencing.
Yes,
So I know, but like not everybody heard you. So I'm repeating for them. So no, that's okay. That's absolutely okay. I think I have my lecture here. Oh, I wanted to do that. Can you open with, so we can write on it? Okay. So we got to go way down. Let's see.
you get on slide 65.
65. Yeah, but I don't know if this is, it's not numbered for me in this way. So it's down here. Oh, but it is numbered because it's the page number. So, gene, not gene chip. Keep going. Okay. I remember. Gene expression. Here we go. All right. So let me zoom in. What? Oh, it opened a separate thing. Okay. Bear with me on this. All right. So let's go over next generation sequencing real quick, right? So you start, and y'all can work on this, y'all can listen to this. The question was just about how next generation sequencing works. So you can listen and do whichever you want, right? So what we're looking for is mRNA expression patterns, right? That's our goal. So we isolate just the mRNA from whatever condition the cells are in, right? You fragment that mRNA, make it small. Then we're going to convert it to cDNA, right? We take that cDNA and now we can amplify it in a PCR, right? Now you have a lot of the same things, yeah? Once you sequence all of those different things, then you can do a blast alignment using the alignment tools on the NCBI website. And you can look at the levels of different transcripts, because not every protein sequence in the DNA makes the same protein, right? We can have exon skipping, we can have all kinds of different things happening when you do the RNA processing, right? Okay, so what's your question?
So, like, whenever they're fragmenting the mRNA,
Uh-huh.
is it being cut where they think it's going to match up with those exons? Or
Nope.
is it random?
It's just random. It's kind of like a shearing thing. You just randomly. It's completely random. It shouldn't matter. That's the point. It shouldn't matter. But it's mRNA, so you're only going to ever see exon sequences. You're never going to see intron sequences. The
So
thin
like
thing
somehow
here,
it's split.
right, the thin thing here is saying that we had this mRNA sequence and it was here, and then we had this mRNA sequence. So it's got to map, because it's mRNA, we don't have the introns. They've been spliced out already. So that's what the thin bar means. Yeah. So the question was, is the cleavage, the fragmentation here specific, or is it random? And the answer is it's random. And whether or not you're like looking for a particular exon, and you're not. That's the whole point of this process, is that it's non-specific. You have no pre-designed assumptions. We think this protein's going to be there, so we put it on a chip. Right here, we have no idea, so we're going to sequence everything that we get and then just map it and see where it maps to. Anybody else have questions on any other procedure that I went over in this section? Because I know that was kind of dense. Okay, I'm going to try and be quiet. And if anybody has a question, ask. Okay, and now, okay, you have a question, but not about the material. If it's a question you want everybody to see, that's cool. Do you have an idea? Oh yeah, I did want to talk to y'all about this. So the question is, do you have an idea for what this class will do for makeup Saturday classes? So we will not be meeting on campus on the weekend. I have an 8-year-old. It's not happening. So I have selected 2 dates. I need to send them out to y'all. So it'll basically be like an extra lecture.
Is it
And that's it.
required to attend, like what if we have to?
It is asynchronous, which means it's just another pre-recorded lecture that you watch on your own time whenever you can make it work. I know it's important to be flexible. I get that. So I know y'all have work. I know all that kind of stuff. We're not going to meet for class over the weekend. We're not doing that. So you know how I normally post your pre-recorded lectures to watch, and that's your homework? One weekend, I'm gonna post you an extra, and that's gonna be our in-class assignment, right? And then another weekend, I'll do it again. So it's just an extra video to watch over what you normally do. So you won't have to meet at any crazy time. You won't have to participate on Zoom. It is asynchronous. Do it when you can. And I'll make sure it's included in the class calendar. Like this is an extra that's due. We're focusing our time on a certain set of materials. Like, oh no, don't do that. Don't do that. You need to read the textbook. You need to read the textbook. If I lectured on every single word that was in the textbook, your lectures will be really long, but you need to read them. You need to read it. I'm going to focus more on the stuff that I covered in lecture. Absolutely. But it's fair game if it's in the book. Do I know what the lab is doing? Jocelyn, in the lab, on the week of spring break, not spring break, fall break, we didn't have an in-class lab planned. And so what we're just going to do is have an in-class lab on that week. And that will make up the day we missed. So it's not a big deal. Group A today. Oh, that's another good question. Group A today. Where'd I go? Here it is. It's going to go to Moodle. And go down, go down, go down. Okay, here, Labster, Lab 1, pipetting, mastering the technique. You have a Labster lab. I did everything I was supposed to do, and I'll show you something. When you click on it, oh, now it's working. This is exactly what should happen. It should say, welcome to Labster, select an option below to sign in. You're gonna sign in, and this link puts you in my classroom. So when you follow this link, it is going to allow you to make a Labster account, and it will allow you to start working on this pipetting mastering the technique simulation. So this is your first simulation. I'm giving it to both A and B. I know B, we're meeting in the lab later this afternoon, but I'm giving it to you both so that we can get kind of caught up. And then A, next week, we'll be doing spectrophotometry, and B, next week, we'll be doing some solution preparation, okay? So we're gonna start alternating instead of going 2 weeks between labs, okay? Can you tell us anything about the format of the exam? You will have multiple choice and you will have free response. So it'll be a combination of the two. Majority multiple choice, but there will be discussion, good discussion. Like, you need to discuss. Like, you know, every time like we answer a multiple choice question, I'm like, but why? But why, right? Morgan, I know we were working with some technical things going on. It should be, you should be getting it today. I'm gonna go check on him again when class is over and see if he's got it up and running. My Labster says, well, why does it work on my computer and not y'all's computer? Because I just clicked the link. Why are you snitching on him? because I help take care of him. So I need to know so I can help. All right, Paige, can you try it again? Yeah, well, Labster has been, you tried it a few times. Can anybody else try it right now who's in my lab and tell me if it works, or is it just Paige who's having? No, Caitlin's also having. So other people try it and tell me what happens when you try it, because on my end it looks fine. Do I miss Edie White? Of course I miss Edie White. Do you, Cameron? You don't? Oh, Ethan, you don't? Oh, but you missed me, right? Annie and Ethan and Cameron, y'all missed me. She doesn't miss you yet. I don't believe that for a second. See, Annie's got my back. It's asking to set up a free trial. So with the link that was provided, it should automatically give you access to my class. Mine says, why is it working for some people and not working for others? Oh, try the browser you're in. Check the browser. Try Chrome or Firefox. Try Chrome or Firefox. You're in Chrome. Let's see if anyone can get all the way in and access the simulation.
Do you and Dr. Sept have two different accounts on the?
No, we have two, yeah, we have two different accounts. So I can't see, like, I can't give you access to his and he can't give you access to mine.
You have it accounts?
Yeah,
For the Google Drive, for the wire and lab.
yep.
See, I'm not in the lab anymore. I took it.
So nice, right, that you got that over with. Well, you didn't have to social distance.
Yeah.
It's hard to be in lab and social distance.
Yeah.
Yeah.
I have inorganic lab today. And he was trying to make us, at first what he wanted to do was, he was like, well, you'll come in person and do the lab, and your partner will be on Zoom, and they'll tell you what to do. I'm like, what? I'm like, no. I had to ask everybody. I'm like, are all of y'all okay with coming here? Because I'm not trusting my partner to do an experiment without me, and I don't think they will trust. I'm not doing that.
Yeah. All right, so Ethan's having problems. Colin's having problems. Okay. Well, I am going to call Labster this afternoon. Apparently, that's what I'm doing this afternoon. In my like 45 minutes between class and lecture, and lab. Chew them out, yes. No, you be really, really nice, and then they want to help you. Be really nice. The more problems you have, the nicer you need to be. Mine's not working. Okay, Timothy, I am going to call them. And say, tell me what I have to do. Anybody who got got to this page here able to get in? Because a few people said they got to this where it was asking you to make an free trial alley. It's not working. What does that mean? It's not working. Can you guys well, what do you mean, Allie? It's not working. I got to that page, but no login worked. Register. You have to register because you've never used it before. Allie, a page. Or you can make an account, but then when you click the link, it brings the black page that says license expired. Okay. Okay. So you can make an account. Put my password, the same thing, black page. Okay. Well, I will send y'all, I will post something on the announcements in Moodle as soon as I get information from Labster. Because my license cannot be expired. We just got it. And we spent a lot of money for it so that you wouldn't have to. So, yeah. Oh, I guess I can close the attendance. I'm hoping everybody attended. So I thought one thing that I could do, let's see, go back here. For y'all is to write when I'm gonna check these discussion questions so you know if they're done or not. So I'm gonna edit, no, just gonna do this. Oop. Fill this one in. Dr. B will check on Monday when Charles goes to bed about 8:00 p.m. It's all around. I'm gonna put an approximate. Let me put it in approximate, because if he ends up staying up late, y'all like, Dr. B, what'd you do? Approximately. I'm not gonna put Charles's bedtime, but you know, approximately 8:00. And then once I have it checked, I'll write Dr. B has already checked. That way you know it's done just from the move page. You tried on Chrome, still license expired. Okay. I'm sorry, y'all. I'm so sorry. License expired. Yeah, I'm gonna talk to them. Okay, so y'all were able to register. It's not the registration, it's my license. Okay. I'm gonna be super nice. You know, that's what my mom says, you kill them with kindness, right? Anybody's being mean to you, you kill them with kindness. It works. They don't know what to do. They don't know what to do. You're so nice to them that they have no idea what to do. It works.
It's Dr. Smith
Oh.
expired too.
What?
Is Dr. Smith expired too?
I don't know.
Don't know.
I don't know. We got 2 different licenses. We do have 2 different licenses. So maybe his works and mine works. No, no cyanide. Ross, no cyanide. What did we do at the discussion? Go and help out another slide. Jaslyn, go and help out another slide. Go check somebody's work, go help out. Because what I would like for y'all to do is to look at other people's slides before I look at them. My mom says complain so much they have no choice but to comply. Well, I won't say that I don't pester, but I do it very nicely. Very nicely. Put a big smile on your face.
Domino's pizza and I was at a friend's house and we had a little get together and they came bring the pizza. Well, the pizza was like flipped over, everything everywhere, and so they were all mad. All Domino's, and I was like, "Sir, I'm really sorry to bother you, but this just got our pizza." No, they came back and we brought 2 large pizzas for free. Like they came fixed one and they got us another.
You know, people, especially in the service industry, how many questions should we expect for the exam? I'm still narrowing it down. I have this massive exam and I know that it is too big. And so I'm working on cutting it down. So I don't know yet. I'm still cutting down. So you want me to continue to cut down. But people in the service industry get chewed out so often that when you can you cut the hard questions? No. Well, I am going to have to cut some of them, but yeah. When you're nice to them when you have a problem, it stands out among the rest, and they usually end up giving you something extra just for being so nice. Just being nice. Can I get a preview of the exam? Yeah, on the day of the exam.
So are we going to get a time?
Yeah, it would be a timed exam. So you'll have 60 minutes, an hour, whatever our normal practice exam for sure. Y'all, do I not practice? Do I not post a little practice every single chapter? Oh, that is sort of a practice exam. It's no discussion, but what do we do in class every day? We discuss. I wrote that. I 100 those already. You checked them out. Good, good, good. The first test is always scary. It is. I know. I know. No, self-checks do not count for a grade. Not for me. I'm a different breed. No, they cannot count for a grade. They will not count for a grade. I think it's important for y'all to be able to have low stakes assessments of yourselves. I think that's super important. So that's just kind of the teacher I am.
I like to at least have it.
Yeah, if this is a freshman or a sophomore level, participation absolutely. Y'all are juniors and seniors. Exams. I'll tell y'all a little story. We got like 5 minutes. People are working on it. So when I took biochemistry in undergrad, I took it as a senior, and the professor who gave it he didn't use a book. He had this like set of slides that he liked, And he had, I don't know where he had compiled them from, whatever. So you had to find somebody who had taken the class before and say, can I borrow yours and go make color copies of it? And it was expensive, right, to make color copies versus, because it wasn't on Moodle. I mean, you know, I'm so old. And so you had this binder of slides that you got from somebody else. And I remember the final exam, he walks in and he's got this massive stack of printer paper. And he like slaps it down on the desk. And he turns to the board and he writes, starvation. Go. That was the final exam. You had to take printer paper and you had to write. And he expected you to write for 3 hours. It was a 3-hour exam. Tell me what you know about starvation.
Holy moly.
Did I pass? Yes, I did pass. I made an A. I loved it. I loved it. I was like, let him go. Oh, it's my story. I was like, oh, yes. Oh. Oh, man, I didn't look up. I didn't care what else was going on. I was just, no, I'm not going to do that. I promise. Yes, C's are okay, but I loved biochemistry. And so I just was going to town. Yeah, yeah. History's not so bad. History's not so bad. I like history. I like not making the same mistake twice. That's what history teaches us. Don't make the same mistake twice. Yeah. All right, so it's 11:42. If you're got your slide done, you can go early. But I need you to check the other slides. If your slide is not done, you have to stay on until 11:40. What is it? Oh no, we close at 11:40, not 11:45. So y'all are free to go. I can't even. It's the audio. But if I don't turn it up, I can't hear anybody if they ask a question. Okay, so if y'all have any questions, let me know. But I think y'all are good to go. I just get sidetracked. I forget what time our class ends. I get so excited and I like it so much, right? Okay, so I'm gonna mute